Welcome to our biology lessons مع دكتور هناء. Today, inshallah, we will solve with each other paper 6, March 2025. Question 1. Benedict solution is used to test reducing sugar. Glucose is a reducing sugar. A student estimated the concentration of glucose in a solution. The student used this method. Label 6 test tubes, 0%, 0.5, 1, 1.5, 2, and unknown concentration. Complete the table by writing the volume of 2% glucose solution and distilled water needed to make 4 cubic centimeter of 1.5 glucose solution. In front of us, this percentage concentration of glucose and volume of 2% of glucose solution per cubic centimeter and volume of distilled water by cubic centimeter. And now we have to know what is the volume needed from the 2% of glucose solution per cubic centimeter when we make a concentration 1.5. So if you concentrate well, we can get this volume by cross multiplication by this way. So we can say this 4 multiply 1.5 divided 2, which is equal 3. So we can write here 3. What about if you would like to make the total volume is 4? So 4 minus 3 equal 1. Now this is the volume of distilled water to complete the volume to 4 cubic centimeter. Use a syringe to put the volume of 2% of glucose solution shown in the table into the test tubes 0.5%, 1%, 1.5%, and 2%. Use the same syringe. أول ما نلاقي same syringe, so it's a kind of error. Why? Because this will cause contamination and change the concentration of the solution. So use the same syringe to put 4 cubic centimeter of unknown glucose solution called U into the test tube labeled U. Use a clean syringe, so this is a way of contamination, to put the volume of distilled water shown in the table into the test tubes 0%, 0.5%, 1%, and 1.5%. Use also a clean syringe to put 4 cubic centimeter of Benedict solution into each of test tube labeled 0%, 0.5, 1%, 1.5, 2%, and U. Put the test tubes in a hot water bath at 80 degrees Celsius. This is the condition for the Benedict solution to work. And start the stop clock. Wait for 5 minutes. Why are they waiting for 5 minutes? For equilibrium. What's meant by equilibrium? To allow the solution to acquire the same temperature needed in this reaction. Remove all the test tubes from water bath and record the color of the liquid in test tube labeled 0%, 0.5%, 1%, 1.5%, 2%, and U. And now this is the data written by the student. What about the color that changed in front of us? This 0%, the blue color, this means that there is no reducing sugar because this is the same concentration of color of the Benedict solution. And if we return back, so we can find there is no glucose solution, but just distilled water. So we can use this test tube as a control. Why? It is a control to compare the other result and also to know the effect of the concentration, different concentration of glucose solution and its result. 0.5% give us green and 1% yellow, orange 1.5, red 2. This gives us the graduation according to the concentration. This means that the least concentration is green and the highest concentration of glucose is a red. The unknown solution, so the product or the result is orange yellow. So this means that it's concentration in between these values. Not 1 and not 1.5. So if you just predict the value of orange yellow, so maybe 1.1, maybe 1.2, maybe 1.3, maybe 1.4%. It does not reach to 1.5 because 1.5 is orange and 1 is yellow. So the orange yellow, so in between 1 and 1.5, so the values range from 1.1 to 1.4. Prepare a table and record the result. So in front of us, the data, the percentage concentration of glucose, and the color change. So we have all of these data in the note. We have to put it in a table about the percentage concentration of glucose and the color change, and we have to write it. And also we have to add the prediction of the percentage concentration of glucose. If you just wrote it 1.1, accepted, 1.2, 1.3, 1.4, so all of them are accepted. And next question is asking about to estimate the percentage concentration of glucose in the unknown glucose solution. I just choose 1.3. You can choose from these value 1.1, 1.2, 1.3, and 1.4. All of them are accepted. State the variable that were kept constant in this investigation. We just put the same volume of glucose solution and same volume of Benedict solution, and also the time left in water bath, that the time for equilibrium. Identify the possible source of error present in step 2 and 3. We have to return back to step 2 and 3. Step 2. Use a syringe to put the volume of 2% of glucose solution into the test tube labeled 0.5, 1, 1.5, and 2. And then use the same syringe that just we mentioned before. This is the usage of the same syringe that's cause contamination that change the concentration of glucose. That's making overestimation or increase the concentration of glucose. So using the same syringe that cause contamination, its effect that the concentration of unknown will be higher than it should have been. Next question said, it contains a protein called albumin. Albumin will turn from cloudy to clear when it is digested by pepsin. It is a protease enzyme. So the pepsin enzyme is a kind of protease enzyme. So this happens also inside the human body. So when we want to keep the temperature, so we have to use a thermostatically controlled water bath at 37 because this is the body temperature. Plan an investigation to determine the effect of pH on the digestion of albumin by protease enzyme. So the effect of pH, this is independent variable or the changed variable. How can we change it? So by using buffer solution. Now, how can we change the pH? What are the degrees that needed to change the pH? So you can choose 3 different pH. One of them is acidic and the other neutral, and the third one which is neutral, to be sure that the best pH or the effect of pH on the rate of digestion of albumin by protease enzyme. So at the beginning, we have to bring 3 test tubes, put in each 5 cubic centimeter of albumin of the same concentration and label them A1, A2, A3. Why we need to label them? Because we said before, if we have any experiment related to the enzyme, it is preferable to put the enzyme in a separate test tubes and all other components in other test tubes. And we have to put all of these test tubes in thermostatically controlled water bath for equilibrium, that help all the solution to acquire the same temperature needed, which is 37 as we said before. And then after that, we have to put the buffer solution, a certain volume, for example, 2 cubic centimeter, but at different pH values, 5, 7, and 9. So why we choose 5, 7, and 9? Because 5 is related to acidic pH, and 7 which is neutral, and 9 which is alkaline. And then We have to bring 3 other test tubes. We said that we have to use the enzyme in a separate tube to put all of them in separate tubes in thermostatically controlled water bath. So we have to bring 3 other test tubes and put in each 2 cubic centimeter of protease enzyme of the same concentration and label them P1, P2, and P3. After that, we have to put them in thermostatically controlled water bath at 37 degrees Celsius and leave them for 5 minutes for equilibrium. We said that why this time, or what's meant by equilibrium, to help all of these test tubes to acquire the same temperature which is needed, that is 37 degrees Celsius. And now we have to start to do the experiment by pouring the components of P1, which is protease in a test tube 1, to A1 that includes the albumin by a certain pH, and P2 to A2 and P3 to A3, and check well with the same intensity for 3 seconds. All of these steps and methodology of the experiment and all the control variable and dependent variable will be measuring the time taken for the albumin to become clear using stopwatch and use colorimeter. Why we need to use a stopwatch to measure the time taken? Why we need to use colorimeter to check the clarity of the albumin? It was suspended or cloudy, and we want to change it to be colorless, so we have to use colorimeter or color chart to stop taking the time. After that, we have to repeat this investigation 3 times for each pH. And finally, we have to wear gloves, goggles, and use tongs. Gloves and goggles because we use chemicals and enzyme. Why we need to use tongs? Because we use thermostatically controlled water bath. Next question, describe how to do emulsion test to show that the fat is present in a food sample. We have to use ethanol, add water, shake well. So we have ethanol, water, shake well. Ethanol, water, shake well. So if the white emulsion, if there is a white emulsion, so there is a fat. Next question, this is photograph of a leaf from an oak tree. Draw a large diagram of the whole oak leaf that's shown. Right. This is one of the leaf. How can we draw it? We have to make an analysis for this figure. Firstly, we have to start to make it easier to draw it and to choose the size of the leaf to be bigger. And you have to use much of the space below to draw it. Firstly, we have to draw step by step the midrib, main vein in the leaf, which is double line. Okay. And then we have to try to make the outline to know the details of the leaf. So if you have any problem, you can erase it, no problem, and return to continue back. And I just follow the same steps in my drawing. And after that, I will show you my final drawing. Okay, this is the main one and the outline. Then we can find veins, smaller, thinner. Okay. You have to know this is distorting this vein from different position or from the same position. And you have to concentrate about this leaf has loops. After that, we have venules, the smaller and thinner. I just make it randomly. thinner in its line, like this. Okay, it's no problem. If you have any problem in something that get it outside, you can erase it. You have to use sharp pencil and eraser. Let's see my drawing. This is my drawing at the end. You have to be sure that the drawing is larger and take much more space in the space below. Right. Next question, line PQ on the figure 2.1 represent the width of the oak leaf. We can find this is 5.8 centimeter, but we have to change it into millimeters. So the rule said 1 centimeter equal 10 millimeter. So 5.8 multiply 10 equal 58 millimeter. And then calculate the actual width. And they put for you this the rule, but some people, they are confused about this rule. So you have to know the magic triangle. What is a magic triangle? The magic triangle said, I am. I am. And now we want to measure the actual width. So you just circle it, and you have to know what is the second part of the rule. So I divided M, I, which is the length of PQ that we measured, and divided the magnification power. We can find it below the figure. This is 1.4. So we have this 58 divided 1.4, and they needed to the two significant figure, so it will be 41. Next question, this figure shows a photograph of oak leaf and hollyhock leaf, and a magnification of both is not the same, okay? So give a visible difference between the oak leaf and the hollyhock leaf shown in the figure. Complete the table to give two other visible difference between the leaves shown in the figure. Don't include the reference to the size. We have to look at and concentrate about it. So if you concentrate well, we can find this is the outline is smooth, and this is one is rough, okay, or like the zigzag or jagged edges. Okay, so we have here smooth and the other is jagged edges. And if you concentrate well, we can find the veins started from the 1 point. Okay, from 1 point like this. But this one, not from 1 point, so we can find them. They started not from the same point. And also we can find this loop-shaped leaf with a different width, and the other, there is no loop. So we can find this loop, and another loop, but this one without any loops. Here, no loops. Here, there are loops. Student investigated the effect of light intensity on the surface area of leaves of soybean plant. The first part is 100 soybean seeds were planted in a pot and put in the shaded, which is low light. 100 soybean seeds were planted in a pot and put in a full sun. The soybean seed were allowed to germinate and grow for 30 days. So we have this is control variable and this is control variable. Okay, after 30 days, 3 of the oldest leaves and 3 from the youngest leaf were removed. From each plant, the surface area of each of the removed leaves was measured. So here we have to know which one of them is dependent variable, something that we will measure, which is, they measure, they said it at the last word. So they measured what? So the surface area of the leaf. This is called dependent variable or the measured variable. State why the student used a large number of soybeans, so they are 100 soybean. When we choose a large number of whatever the sample, so to get representative sample or to avoid bias or to identify the anomalous result or to get reliable result, all of them are accepted. You just need one of them. The student estimated the surface area of each leaf using the graph paper as shown in the figure. Suggest how the student used the graph paper to measure the surface area of each leaf. How can we measure the surface area? We just bring the leaf and put it on the graph paper. And then we have to draw the outline around the leaf using your pencil. Then start to count the number of squares on the graph paper that covered by the leaf. Okay, so if you try to make it again here, to make it like as a transparent, I just need to try to make it well. Just like the draft. And then we have to draw one of the leaves, for example. Okay. This, after putting the leaf, we just make the outline around it on the graph paper and then remove the leaf and start to count how many squared are full. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. And then after counting the fully covered squared, and we have to search about the half and above covered. So for example, this one accepted, and maybe this one, but we never accept this one. No, 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 no. Okay, so this is the way to measure the surface area of each leaf. So how can we write it? So draw the outline around the leaf, count the number of squares on the graph paper covered by the leaf, count the squares that are more than half full, maybe half and maybe above, and then you have to use the suitable measuring unit of the area, this one which is a millimeter each square, so the area should be millimeter square. Next question, the result of the investigation are shown in the table. We have the lightening condition, age of leaves, and mean surface area of leaves by centimeter squared. And we have two data. We have two full sun, okay, and we have two shaded area, so it means less light. And we have two young, and we have two old. On the grid, using the data in the table to plot a bar chart to show the result of the investigation. Now, how can we draw the bar chart includes two data and what we are going to write on the x-axis and y-axis. So in front of us, two conditions. So which one of them that will be drawn on the x-axis or write on the x-axis, and which one that on the y-axis? Mean surface area of leaves, we know that this is dependent variable, so we will draw it on the y-axis. But what about the x-axis? The x-axis will use these two data اللي هم مين؟ x-axis اللي هي lightening condition and age of leaves. How can we write it? Step by step with each other. Here, if you concentrate well, we can find this graph paper, this graph question with the 4 marks. So we have to deal with, firstly, axis, label. Number 2, scale. 3, bars. Okay. Right. We have lightening condition and age of leaves. And age of leaves. So we can write it like this. So the first one, we will write full sun young with the data 52. So full sun and young والداتا بتاعتها plotting 52. This one. And what about the next? Shade young with a 45. So we have to write on the x-axis shade and young, and we can find it 45. Next, with full sun old 78. Full sun old, the largest one, which is 78. And the last one, which is shade old 58. So we can write it, shade old, and we check it if it is 58. Next question. The student calculated there was 15.6% increase in the size of the young leaves when they were grown in full sun compared to in the shade one. Calculate the percentage increase in the surface area of the old leaf that had been grown in full sun condition compared that old leaf that grown in shaded one. So the data, yes, we have these data, okay. So we have the rule, which is final minus initial divided initial, multiply 100. Okay, so 78 minus 58 divided 58 multiply 100 equal 34.4. So we need it to one decimal place. This one, I think that this is, it was 48. So the value is 34.4. State to conclusion from this investigation. If you return back to the graph, so we can find that the plant that grow in full sun and old give the larger than the young leaves. So the old leaves are larger than young leaves, and the plant that grown in a full sun have larger leaves than in shaded area. Next question, hydrogen carbonate indicator used to test the presence of carbon dioxide. The aquatic plant was placed red hydrogen carbonate indicator and put under the bright light. The plant takes carbon dioxide as it photosynthesized. State the final color of hydrogen carbonate indicator. We know that an hydrogen carbonate indicator tests the presence of carbon dioxide. If it is more, so the color of hydrogen carbonate indicator changes into yellow. But in this question, they ask about after the consumption of carbon dioxide, so there is no carbon dioxide, so they ask about the negative result of hydrogen carbonate indicator, which is purple. Again, hydrogen carbonate indicator used to test the presence of carbon dioxide. An aquatic plant was placed in red hydrogen carbonate indicator and put under bright light. The plant takes carbon dioxide, so it consumes all the carbon dioxide that exists in photosynthesis. So they ask here about the negative result of hydrogen carbonate indicator of carbon dioxide. They need a negative. We know that in the positive result of hydrogen carbonate indicator is yellow. What about the negative is purple. So the negative result of hydrogen carbonate indicator هتبقى ايه؟ هتبقى purple. If another question asks about the positive, it means there is carbon dioxide, so the color will be yellow. But in this question, in indirect way, after the consumption of carbon dioxide in photosynthesis, they ask about the color of hydrogen carbonate indicator, This means that the negative result يعني this means that there is no carbon dioxide anymore. I hope إن احنا نكون اتعلمنا حاجة جديدة النهاردة، وأشوفكم إن شاء الله في الامتحان اللي جاي. ما ننساش نعمل لايك وشير وسبسكرايب علشان يجيلنا كل الفيديوز الجديدة.